Good afternoon, everyone. I'm pleased to see such a great turnout on a fairly wintry day here in St. Louis. We ordered this specially for our visitor from San Diego, just to make him feel like what the rest of us have to endure. It's my great pleasure today to introduce Steve Mayfield, and this is a joint PARC and Plant Lunch seminar. And I will also remind you that Steve will be giving a second talk tomorrow afternoon at 4 p.m. in biology. Jeremy, what's the room number? Rebstock 322. So that will be a second talk on a slightly different topic. But today, Steve is going to tell us about synthetic biology to improve the production of bioproducts in algae. Steve is from California and got his PhD degree at UC Berkeley, working with William Taylor, and then went to Geneva on a postdoc with John David Rocher. And then after that, came back to San Diego, where I guess he's been ever since. Is that right? And he was for a long time at Scripps Research Institute, and then in 2009 moved to be a professor at UC San Diego, where he is now the director of the San Diego Center for Algae Biotechnology. And one of the things that Steve has worked on his whole career are genetics and molecular biology of algae. And as time goes on, his interests have very much centered around the use of algae in bioenergy applications. And to that end, one of the other things that he has been very productive at is starting companies and being entrepreneurial about his work, which of course is extremely important in the bioenergy area. And he founded Rincon Pharmaceutical Company, and the one that perhaps many of you are knowledgeable about is Sapphire Energy, which is one of the premier biofuel. And so he has quite an experience both in the scientific realm and also in the business arena. So Steve, welcome to St. Louis, and we are very happy to have you here.
Thanks very much.
Oh. On exploiting diversity in synthetic biology for the production of algal biofuels. So I really recommend this to you if you're interested in this area. So.
A good little bit of background. Bob, thanks very much for the invitation to come out. I'm going to tell you about a couple things today. All of the stuff today I'm going to talk about is how we could use algae to make bioproducts, and mainly recombinant proteins. Although I'll tell you a little bit about what I think we can use algae to improve photosynthetic productivity, which could then translate over to higher plants right at the end. Sort of some things that we've done in some theoretical stuff. Okay, and then tomorrow I'm going to tell you a little bit about bioenergy, energy in general, and then a bit about that today. Okay, I do have one question. How do I advance the slides now? They took my laptop away, and I've got a screen. Does anybody know? Are there any 15-year-olds in here? They can probably do it with their cell phone. Oh, the keyboard down there. Okay, got it. Ah, excellent. Okay, good. Okay, so why do we need to improve bioproducts in algae? Why do we even care? Ago, we were food sufficient, right? We could produce enough food to feed the planet, no problem at all. Where we had problems on the planet was because of distribution, not because of general productivity, right? That was brought about by something called the Green Revolution, which really started back in the 1950s and revolutionized the efficiency of producing food, all right? And so this happens to be wheat yields in India, but this could be corn. This could be anything worldwide. And it's just tracked up really well. We sort of hit a peak about 2000. It's sort of leveled off a little bit. But we of two things, really. Mechanization. So we started using tractors and less hand labor on it. And fossil fuel, petroleum use. So because we were driving tractors, but also because of the huge amount of fertilizer we put into these things, right? That was a huge energy input in. So on a per person basis, on a per capita production per farmer, it went up enormously. But in terms of energy in versus energy out, it actually wasn't a very good deal. So as many of you will know, fossil fuel accumulated on this planet for about the last 400 million years. And we're going to burn through all of that in about 200 years. So that's about 1,000,000 to 1. 1,000,000 years of reserve for every 1 year we're going to burn it. And I think everyone can kind of have a feeling that that's probably not sustainable, all right? And that's starting to show up now. And as I said, we'll talk about energy tomorrow, but that actually becomes very important when we think about all other products. As our fossil fuel decreases, the price of it goes up. Now, when fossil fuel was really cheap, which it was between 1950 and 2005, it was not a large component of what we did in agriculture or in any bioproduction. Right? In other words, when fuel's only $10 or $15 a barrel, it's not a big enough percent of food to really count. But as the price of that goes up, and really here starting in 2005, our first big spike in oil came in about 2006, and right along with that, so here's our spike in 2006, hit $150 a barrel, and the price of corn, but this could be any commodity, went right with it. All right? Then the economy crashed, price of oil went down, price of food went down, now the economy's coming back, and back it goes. And the reason for that is because now that oil or energy is expensive, it is now a larger component of food production. But this is true for anything, okay? This is true even if you talk about fermentation in bacteria or yeast. All right? They become more expensive. Okay. The biological conversion of sunlight energy into chemical energy. So fuel, petroleum, is ancient algae, and coal is ancient plants. So all of the energy we consume, I mean nuclear and geothermal, and there's a few others, but all the rest of those are brought about by sun. All of the liquid fuels, all of the chemical fuels we use, that's all photosynthesis. All right? So what we're doing is we're taking ancient photosynthesis fuel, and we're converting it into plastics or any bioproduct we need. But we can also do that in real time, right? So we can take photosynthesis and we can convert it into products. That's what corn is. That's what food is. But you can convert that into any product. You can convert that into carbohydrate. You can convert that into protein. You can convert it into lipid, right? So we can make anything we want. And the real advantage of photosynthetic organisms is the energy input is sunlight, not fossil fuel. And in fact, you can take the lipids out of here and turn those into fuel. We'll talk about that tomorrow. That's what the idea behind Sapphire, between all of the bio companies that are trying to make biofuels. Okay. So we think about algae as a photosynthetic platform or as a primary energy platform for a couple of reasons. First and foremost is productivity. All right? It doesn't do us a whole lot of good to do bioenergy or bioproduct production if we're doing it in efficiency or if we're doing it at the cost of agriculture of food now. So number one, we have to get new productivity. We can't just convert corn, into ethanol and burn that in our tanks and think that's a gain, right? That's the food versus fuel debate. But we also have to think about this as there are now 7 billion people on the planet, and all of them want to live at the same lifestyle that we do, and that means they all want to consume, or they're all going to consume the same amount of energy we are, right? So either our energy can find much more efficient ways to do stuff so we can maintain the same standard of living in a much more efficient way. And that's really what productivity means. What productivity really means is how efficient can I get? Now, we can measure this in a lot of different forms. The easiest is probably just chemical energy, biomass. So how many tons per acre per year do I produce? And on that front, algae is the most productive organism on the planet. There's a couple reasons for that. One is that during most of photosynthetic evolution, the world had very high amounts of CO2. all heard, because the CO2 is increasing, climate change is increasing, but really for the first couple billion years, CO2 was at very high levels. And so the enzymes and the whole process that evolved in a very CO2-rich environment, and so those enzymes are not very efficient at capturing CO2. All right? But because algae grow in water, we can actually increase the concentration of CO2 in the water dramatically above what it is in the air, and therefore we can drive that reaction. So many of you may have heard that there's something called the C3 plant and a C4 plant, and C4 plants are much more productive than C3 plants. That's because they have a carbon concentrating mechanism. They have a way to concentrate CO2 near the primary enzymes that fix it. In algae, what we do is the same trick, except we have the water as that concentrating mechanism. So we can hit unprecedented productivities with this. You have to have a scale that you can go to that's meaningful. Right? In this country, we grow 95 million acres of corn, 56 million acres of soybean, 45 million acres of wheat. These are enormous numbers. We burn 300 billion gallons of petroleum every year. So you can have the best process in the world, right? And if you can't take it to the billions of gallons, billions of pounds, if you can't take it to that scale, then it really doesn't matter. You're not really going to impact what it is we have to do. You could be a very profitable company. You could have a good research project, but you're not really going to impact the world. So you have to have scalability, and I'll show you a little bit about that at the end. Sustainability is a funny word. Well, it's not a funny word, but it's a funny concept that people use. What does that really mean? What that really means is, like I said, it doesn't do us any good to produce energy in algae if we're going to consume up all of our nitrogen and phosphate in order to do that, and therefore not have enough of those to grow the other crops. So it has to be something that we can do every year for many years to come. And then the last thing that I think, and we'll talk a little bit about this at the end, is diversity. One of the things that's coming, well, it's already here, but one of the things that all students should be thinking about is how do we're in an era now where I am no longer limited by genetic resources or computing power, right? Or frankly, even my imagination about the organisms that I want to create and what I want to make, right? And algae is part of that because there are estimated to be 3 million species of algae on the planet. They live everywhere. Several billion years, and with that comes a set of traits, a set of genes that we now have the capacity to understand and exploit, meaning to move those from one species to another. The traits, right, and put those into corn, even a fraction of that salt resistance. If we could get corn to grow in 30 or 40 millimolar sodium chloride, I think that's something that's, and it's sort of summed up in this slide. We clone very few of them in my lab anymore. Here's a new genome. Here's a new set of genes. I understand some of these, and I think this is a new metabolic pathway. And here's 3 that I kind of think are probably involved in this pathway and maybe produce this small molecule at the end of it. I simply type those sequences in. I ship them off to a DNA synthesis company. 2 weeks later, those genes come back. I cut and paste those into a transformation vector. I put them in, and then I measure the output to see if I was right. And today, we're wrong more than we're right. We're probably wrong 90% of the time. But every time, that gets better. And as I understand genomes better, and as I understand DNA better, my error rate is going to go down. And so that in the next few years, maybe 4 or 5 years is my guess, science will be done in silico, and then you'll simply prove it. You'll make one final designer organism at the end to prove that your entire theory of gene regulation and the whole system that you developed is correct. All right? So I don't think it's going to be, you know, some people will say, and if you listen to Craig Venter, he says we're going to invent organisms from scratch. I'm going to make designer organisms. I don't believe that to be true. But I don't believe that to be true because I don't see any reason to reinvent the wheel, right? These systems, any photosynthetic system, whether it's cyanobacteria or algae or you name it, spent billions of years figuring out how to do things very efficiently in terms of photosynthesis. So really what we want to do is we simply want to domesticate them in making the whole thing, right? When a new car comes out, you don't reinvent the whole new car. You might put a new component in it, a new engine, a new whatever. But it's an evolutionary, not a revolutionary, not a big jump, right? So algae has this unprecedented diversity in biological traits, but it has another thing, right? So that's our source genes. But the other thing it has is because it's a single-celled organism, and because over the last 20 years in medical biotechnology, there's been an enormous boom in sort of the high-throughput screening things, whether that's robotics for handling single cells, and algae fit into all of those things. They work really well, right? So that means that I can look at 100 million or even 1 billion individual cells in a 50 ml flask and analyze traits for that many. As long as I have good stalking horse, as long as I have a good reporter for those things, I can analyze that many. And so I can do this in a very high throughput way. Then I can think about putting those into the ultimate organism that I want to produce. So I could put those into corn or soybean or wherever I want. Or maybe I could just leave them in algae. And I could make food, and I could make fuel, and I could make products. And some of these things are already on the market. We already sell wrinkle cream from algae. We've been selling human nutraceuticals from algae for years. It's about an $8 billion market. And now we're really starting to make some of the designer products. And those are what I'm going to tell you about today. Some of the designer products that we have started to make. All right? So, photosynthesis takes place inside the chloroplast. This is true in all eukaryotic photosynthetic organisms. In cyanobacteria, they're bacteria, so it takes place in something that looks like the chloroplast, which is what the whole cyanobacteria cell is like. Genes are encoded in both the nucleus and the chloroplast. And in fact, the chloroplast in all higher plants and algae is pretty small, 200 kilobases or so, encodes about 40 to 50 proteins, but has its own genetic material, right? Meaning that it has ribosomes and transcription factors and et cetera, right? But most of the genes are encoded in the nucleus, translated in the cytoplasm, and post-translationally transported in, which means if you want to impact something that's going on in the chloroplast, you can transform either one of those genomes. But they're also biochemically very different. The environment of genes in the cytoplasm or proteins in the cytoplasm is very different than the biochemistry inside of the chloroplast. And I'll show you where we took advantage of that to make some what I think are kind of unique proteins. Okay? So chloroplast transformation proceeds by homologous recombination. Chloroplast came from cyanobacteria, and so their promoters and most of their control elements look very bacterial-like. They're very simple. All right? So the promoters and untranslated regions are the regulatory components of these things. They're relatively simple. You need fewer transformation events to get the product you want. Recombinant proteins can accumulate to pretty high levels. I'll show you some of that. And we can make proteins you can't make elsewhere because of this unique biochemical environment. So as I said, these are the coding regions. There's only about 40 of them. Relatively simple promoters. The kind of interesting thing which is different about chloroplast compared to most bacteria is that the photosynthetic complexes are large 20, 30 protein complexes, and they tend to have relatively structured, complex structured proteins. So chloroplasts have a whole set of proteins for folding complex proteins and for making disulfide bonds, which bacteria generally lack. All right, and I'll show you how we took advantage of that to make some mammalian proteins inside of the chloroplast. So what you need for that for transformation of anything is you need a selectable marker gene. As I said, it goes by homologous recombination, so you need a couple homology regions to integrate it into the genome. You have to find a place in the genome that we call silent sites, which is just a place to integrate the gene that won't disrupt some other genetic function. And then you need a promoter and a UTR, and you can drive accumulation of a recombinant protein. And then we'll quite often put tags on these things just to follow them around. Many years we worked on what are the rules of light-regulated gene expression inside of first in plants and then later in algae. And what we discovered was, unlike bacteria, chloroplasts have very complex 5' untranslated regions, and this sort of seems to be the business end of gene regulation inside a chloroplast. So they have relatively simple promoters. In fact, they don't have very many sigma factors, transcription factors. It appears to be only one in Chlamydomonas, 2 or 3 in some other organisms. But you get beautiful light-regulated gene expression of these genes. One of the things they respond to really well is turning the lights on, and when they do that, you get an increase of translation of almost all of the photosynthetic proteins, and all of that is brought about by these structures. They have ribosome binding sites, but unlike bacteria, those aren't -7 to -10 from the initiation codon. They tend to be much farther back. Almost all of them have some sort of stem loop structure, which is essential. But these are very complex structures, and by making individual mutations into this, we could identify some elements which were required. So we could identify that this stem loop was required, that some of these other loops might be essential. But it's very difficult in a structure like this when it's not primary sequence that is determining regulation. So in a promoter where you have double-stranded DNA, by and large, it's primary sequence, right? If you have a transcription factor binding site, which is 8 or 10 nucleotides, and a transcription factor that binds to that, you can get expression from that. But in RNA, they tend to fold into complex structures, and almost never do we recognize linear sequence. So although we know there are a set of proteins that bind to this, in fact, there's 6 that we've characterized that bind to this structure that are required for light-activated translation, none of them seem to recognize primary sequence. Okay? They recognize some secondary sequence or some fold or some maybe a short primary sequence like this that has also something to do with spacing. Okay? So we were frustrated by this, and we had made dozens of mutations, and we had identified something. In fact, we had made hundreds of mutations to identify a few dozen key components in about 5 or 6 different genes. But then I happened to be up in the Bay Area giving a talk a few years ago at Agilent, and Agilent is the component company, right? They make all the mass specs and most of the equipment that we use to measure things in lab. But they also had developed technology which they call their OLS mutagenesis. So they could make libraries of oligos that were 200 nucleotides in length, and they could print 50,000 of these oligos on a single 1-inch square glass slide. Okay? So that means you could get 50,000 unique sequences in a 1-inch square. And when they originally developed this technology, the idea behind it was, oh, we'll do this like chip technology so that people can measure, on an array, different gene expression. But then myself and several other people realized that, yes, but if we could just cut that DNA off those slides, then we would have a library of unique sequences, and we could start to do true synthetic biology. So not just synthesize an individual gene, but synthesize libraries of genes and then interrogate the whole library in one shot. All right, so the idea behind this was pretty simple. In Chlamydomonas, recombinant protein production is limited by translational structure. All right? So if we could identify specific elements that convert positive or negative regulation, that might allow us to design synthetic UTRs and get around some of the control issues that we have and just learn to regulate the system much better in a truly synthetic way. All right? So we used the Agilent library. We actually ended up making about 3,000 different mutations, and we scanned across 5 different 5' untranslated regions. So this is just one example of the many, many that we designed. This one happens to be 16 nucleotides, where here's the entire 5' untranslated region of a single gene called PSVA. This is one that shows great light-regulated translation. It's the one that I just showed, that complex structure. And so we simply scanned through this thing with A's all the way across. And then we did ones where we did 3 A's and scanned across it, and 6 A's. And we made every combination we could think of. And then we even did ones where we took chunks of 16 and mixed and matched them all around. And so when you do this bioinformatically, you can see that I can get every combination possible that this UTR could exist in, right? Not changing every single nucleotide, but at least down to the 3 nucleotide pairs. So Agilent synthesized that entire library for us, all right? So they shipped it to us as just dried little oligos. Using something called user cloning, we clone those into an expression vector where we replace the endogenous UTR with an entire library of variants across this thing. Put that into a transformation vector, transform that first into bacteria, and then pulled that for the plasmids, pulled that plasmid library out of bacteria, and then transformed that into algae. Okay? With the goal obviously being that what you'll get is variants here. Sometimes you'll get guys that don't express at all, right? Because that mutation that you've introduced into that part of the UTR is a required element and you've killed it. Sometimes you'll get guys that look just like wild type, like maybe one of these guys right here. But sometimes you also might get guys that are brighter than wild type. You might get ones where you've knocked out a negative regulatory element. So we did this, and this was just our first shot in where we actually put this into E. coli. And so one of the shocking things that we discovered right away, we kind of already knew that chloroplast and bacteria were related. You know, we could see that, oh, that promoter element kind of looks like a -16 element and a -35 element, and we see a ribosome binding site there. But one of the really interesting things is when we screened our first library, so we put this entire library in front of luciferase. Okay? Screen the coli on luciferase, and the first thing we found out is, wow, when you take and do a mutagenesis of a chloroplast promoter in UTR and put it into bacteria, suddenly you get some guys that express really well. You get other guys that are dark, and here's just a bunch we picked out. So you get some that don't express at all, and you get some guys that are really bright. So we already knew just from this, A, that our mutagenesis strategy was at least working. We were getting variants, and B, that you could actually turn a chloroplast gene into a bacterial coli gene with some pretty simple mutations. All right? If we had just put in the wild-type chloroplast gene, it would look like this. It would not express in coli. Then we put those things into algae. And we had to get some undergrads because we had to pick about 30,000 each of these algae, and we struck them out. So what we do is they would go and pick 50 per plate and pick hundreds of plates. And it's just, they just have a little streak of algae. So there are 50 different colonies on here, 50 different individual cells. So here's a negative control. Here's an algae with a luciferase gene put in with a dead promoter. Here's the wild type guy. So obviously if you just put in the wild type UTR, you'll get some expression inside the chloroplast. And then here's what you get out of the library. So you get a bunch of guys that look like wild type expression. Of course you get a bunch that you've now killed that UTR. And then you get some that actually express much better. All right? Then, because you have about 3,000, right, our assumption was, 30,000. So our assumption was the library size was about 2,700 different mutants we had put in. So that means you have to have 3,000 for a 1x coverage, but because you don't know how many times you're going to get the same event, like that exact same guy put in, you actually have to pick a library 10 times bigger to make sure you get every Agilent's chief competitor, Life Technology, and through their new sequencing, sequenced about 100 million of these guys. So we just pooled the entire library and sequenced it many, many times over. And what this gives you a way to statistically make sure that you've gotten every single variant in there. And then you plot these guys. And so what you plot is, so here's the analysis of one of those 5' UTRs. So this is from the PS. And what it plots is, if it was 100%, what that means is that every single time you sequenced that nucleotide, right, from 30,000 different colonies that we picked, it was exactly wild type. And what that tells you was that that means it had to be wild type in order to get expression. Okay? For the guys in blue. So these are the ones that had good expression. If that blue line is down, what that tells you is that the tendency was for that not to be wild type. So if I had good expression and this is down, that tells me I must have a negative element there, or have something that I don't want it to be wild type. I get better expression if it's not wild type. Okay, and then the red line is just the opposite. The red line are from strains that didn't express well. So in those guys, you would think that, so what you should get is an inverse line, right? Things that express poorly, if there's a positive element that's required for expression, you would think that every time it wasn't wild type, you would get poor expression. Sure enough, that's exactly what we get. Now, this is comparing one that had been analyzed many, many times by a number of different labs. Right? It was one of the 5' UTRs that got beat up by us and several other labs over many years, and that's what these 3 bars are right here. These are places that previously had been identified as essential elements for expression of that RNA. And when we analyzed our sort of synthetic approach in high throughput screen, what we saw is, oh, we find all of these guys in our library, but then what we find out is some of these elements, so for example, here's an element right here. Previously, people said, oh, we think this is required, and it is. That's the Shine-Delgarno-like sequence, and that's clearly essential. If you mutate that, you lose expression of that is upstream of that thing is also absolutely essential. Right? Here was another element that people said, oh, we think something is required here, but it turned out in ours, we looked at that and we say, God, we really don't see that element. If this was truly a required element, then this blue line should be up here and the red line should be down here. And in fact, over the last few months, what we've realized is all of the mutations that people generated in this had been deletions. And what they were really doing was changing the spacing of that element to the AUG. All right? And then we also identified this element back here. Okay? So what that tells us is what our real goal on this one was, could we make synthetic elements out of this? Okay? So here's another one called the PSAA, 5' UTR. So this is one of the photosystem 1 genes. And it also shows light regulation and control by a lot of different things, iron and a few other things. But what we noticed on that was there were 2 really strong elements right up here at the beginning of it, which appeared to be negative regulatory elements. Elements are there, but they seem to be slowing down translation or repressing translation. So we simply went back and said, okay, we're going to keep these elements. So here's a positive element. Here's a positive element here. You can see the blue lines are high. You can see that they're positive because these red lines are low. Okay. 1.5 or 2 times wild type. All right? So we could actually make synthetic elements that could express proteins at a much higher level. So construction of synthetic UTRs without these elements showed nicely enhanced translation. Now what we've done now is gone back and asked, okay, and now can we do iterations of these positive elements the way you can in promoters and actually enhance translation beyond that? And we don't know yet what that answer is going to be, but my bet is that's exactly what we'll get. Okay, so as I said, you know, we can do this now in a high throughput way, and we can express luciferase, but can we actually make products? And I'm convinced, as I'm sure many people here are, that, you know, the future of science is not just can I make basic discoveries anymore, but it's what problem can I solve? And I started this whole seminar by saying that I think that the biggest problem we have going forward is inefficiency. Right? This is absolutely true in energy. We have to learn to use much less of it. We have to learn to produce it better. But I think this is true in everything. This is especially true, I think, in medicine as well. You know, for many years, we've always had NIH, and all of our research has been designed, make the best drug you can possibly make, right? Make one that's very effective and will absolutely treat that disease with no concept that there's a cost-benefit analysis, right? It's like, how much does that drug cost? There's a new drug, and I'll show you a counterpart that we've made of that, which is an anti-cancer drug, which will be on the market this year. It's going to cost $140,000 for 4 injections, right? And that's simply unsustainable, I think. So one of the things that we've tried to think about is how can we use a photosynthetic organism to produce medicine in a more efficient way? And the most efficient way, I think, that you can produce medicine is if you make it orally available, right? The most expensive part of any drug, people will always tell you, are the clinical trials, all right? But the reason they're expensive is because we have to produce a protein, we have to purify it to unbelievable purity, then we inject it into people, right? And all that part of the process makes these things enormously expensive. But if we could just eat it, right? Number one, we get rid of purification costs, right? It becomes much safer Because when you inject things, you have to worry about, am I introducing viruses, am I introducing bacteria, the rest of the stuff. If you eat it, your body's already designed to take care of that. So it occurred to us, but other people as well, that there's actually already a biological paradigm for this out there, and that's called colostrum. Colostrum is the first milk that all moms make, all mammals make, and give to their newborns. And there's a lot of different proteins inside colostrum, right? And some of them, it's kind of obvious what they do, all right? So some of them, there's lactoferrin. This is a protein that binds iron. Okay, that's pretty obvious. Newborns need iron. That's going to transport it across. There's another one in here that binds it across, right? But there was one called mammary-associated amyloid, or serum amyloid A3, all right? This is found in every single colostrum out there. And a group in Nebraska was going through colostrum and attempting to identify what do all these different proteins do. And some are antibacterial, right? They're quite well known. And this one wasn't well known. And what they figured out was that what this protein does is gets into your gut and stimulates mucus production. And by stimulating mucus production, you get a physical barrier from bacterial and virus. So they don't adhere to the intestine. And that keeps you from getting severe bacterial or viral infections, which is bacterial or viral diarrhea. And many of you will know that that's the #1 cause of child death in the world, right? It's not malaria, although we made a malaria protein. I'll show you that. It's not malaria. It's not AIDS. It's actually just diarrhea from bacteria, okay? So we simply asked, could we express that protein? It turned out we could express that protein really well. Here's wild type. This is just a stain gel showing 2 different strains of that, making the serum amyloid really well. Here's a Western blot of that. We then purified that protein and tested it against human epithelial cells, and we could show that by treating this protein onto epithelial cells, we could stimulate mucus production about 300%, so about 3 times, all right? We then wrote, so we actually did this in 2007 and 2008, and I thought, oh, this is a great candidate because people will really understand that I can now produce essentially a drug, right, in an edible algae that I can feed to people at less than a penny a dose, right? In fact, our calculation was that at 0.4 cents a dose. So you could dose this thing for a whole week for 4 cents, right? And that's what it takes, about 7 days of dosing this in your file. We wrote grants to everybody, and nobody understood it. And we sent one into the Gates Foundation in 2008, and even they didn't understand it. And then last year, they called up and they said, hey, remember you sent a grant in 2007? Yeah, vaguely. And they said, we didn't get it at the time, but now we do. So we will give you, not one of the pigs we use, this guy's a little bit older, but you can tell from his smiling face that the reason it's up there is because they worked wonderfully. So what we did was we took newborn pigs and we let some of them see colostrum from mom. We let some of them see what's called milk replacer, which is what pigs get when they're normally born because you take them away from mom as quick as you can so there's no competition in feeding and you don't get runts, all right? And then we gave some of them algae, non-transgenic algae, just wild-type algae, and we gave some of them algae with our SAA in it. And we treated them all for 7 days, and then we challenged them with one of the infectious forms of E. coli. And of the untreated guys, or the ones that got algae with no SAA in it, 5 or 6 of the 8 pigs died. They actually got so sick that they had to be euthanized. The ones with colostrum all survived, but 6 of the 8 were not putting on weight at the right amount that farmers like. And of our guys, 8 of them put on weight, and then we did a necropsy on them. And of ours, of the guys treated with algae and SAA, only 1 of them showed any sign of lesions of bacteria. Of the guys treated with colostrum, 2 of them showed severe signs of lesions, and the guys untreated, all 8 of them did. So what that means is we now have a drug made in algae, which we think we can treat at fractions of a cent per dose, which would be available for any part of the world in which seasonal bacteria show up that give you diarrhea. So this wouldn't necessarily work on something like cholera, which is specifically designed as a pathogen, but it'll work great on all India, someplace where maybe sanitation isn't so good. And so what you do is you dose yourself with this for about a week before you go. Now, it won't give you protection from a specific bacteria, but that's not what you want because you don't know what specific bacteria you're going to run into. What it'll give you is systemic protection against all of the available, because that's what colostrum is. That's the only way you ever see it. So we think this is an enormous opportunity to reduce the cost of medicine in this country, but also to make it available to the rest of the world, which right now it's not. We have all of these fantastic discoveries in this country, but no drug that sells at $100,000 is available to anybody but a fraction of our population or European population, let alone the rest of the world. And I think this is something that all of you, students especially in this audience, have to think about, right? The old paradigm that I grew up in, which is medicine will be available to everyone and cost is not something to consider, is done. Right? It was never sustainable in the first place, but it certainly isn't going to be sustainable in the future. So we have to think about a cost-benefit analysis, and we have to think about driving efficiencies. How do I make something more efficient? All right? So here's another one. I'm just going to burn through these last couple of guys pretty quick here. This is the anti-cancer drug we made. So why did we make this one? We made this one because it occurred to us that chloroplasts have a translation machinery that looks like prokaryotic, right? And that means they should be resistant to eukaryotic toxins. For whatever reason, most eukaryotic toxins, whether it's ricin or exotoxin A or cholera, they attack the translation machinery of eukaryotic cells. Some of them attack the ribosome, some of them attack translation initiation factor. I have no idea why. It obviously is just a susceptible place, and bacteria have learned how to exploit this. But because the translation apparatus inside chloroplasts looks like prokaryotes, we thought, hey, I bet there's a chance we can make eukaryotic toxins inside of a eukaryotic cell if we keep them sequestered inside the chloroplast. Okay? We had already previously produced antibodies inside the chloroplast. So I had said that chloroplasts were good at folding complex proteins. We made human monoclonal antibodies, heavy chain and light chain, and showed that chloroplasts understand those things. They fold them and they form disulfide bonds correctly. So we went back in and just asked, can we make all of these proteins inside the chloroplast? Because it gives us an ability to make a protein you can't make anyplace else. All right? And so these are just those three different proteins. We could make all three of them. Here's the single-chain antibody that accumulates well. Here's what's called a monomeric immunotoxin. So that's a single-chain version of that, where we put the heavy chains, which form disulfide bonds and link together. So what you get is two binding heads and two toxin domains, right? So twice the binding and twice the killing capacity of that guy. And they accumulated just fine in chloroplast. What that tells you is something really interesting. That tells you not even one molecule leaks out of a chloroplast. So I said in the beginning that most of the proteins in photosynthesis are translated in the cytoplasm and transported into the chloroplast, and they do that. Those proteins that go in, no proteins come out, because if even one molecule of this got out, it would have killed the ADS ribosome inside the cytoplasm of the algae, and they didn't do that. These guys grow just fine. But here's the more important one. So this is killing assays of a human tumor cells. There's two different ones we look at. One is called, the toxin on this one is ETA, but we've coupled several different toxins. ETA is exotoxin A. So what it does is it binds to elongation factor 2 and ribosylates it. Okay? So it'll kill any eukaryote. So what these are is you have an antibody on one end, and in this case it recognizes CD22, which is a surface antigen on B-cell lymphomas. It binds to those, endocytosis in, the exotoxin A knocks out translation, kills the cells very quickly, you get apoptosis. Okay, so here are the three different cell lines. Jerkcat is a T cell, which doesn't have CD22 on its surface. Treat those with our immunotoxins, you get nothing. Here's treated just with a single-chain antibody alone, doesn't kill the cells. Here's the single-headed binding guy, kills efficiently, and here's the two-headed binding guy, kills very efficiently in cells. Then we transplanted Ramos B-cell tumors into mice and then showed that we could cure the mice of those tumors by treating them with this. So this is an antibody toxin. This protein is identical to the one that is going to be sold at $140,000 for 4 injections of it. So we think this is an opportunity, right? Although it doesn't save you anything on the clinical trials, at least it's an opportunity to start to make these things at a fraction of the cost of the way. We made these in algae. The reason you make those things is because they have many disulfide bonds and they cannot be glycosylated. So if you make these things in bacteria, the disulfide bonds correctly, but then they put sugars on them and that masks the antigen, so they're not immunogenic. So again, we think this is one of the few places you can actually make these things, right? Complex fold, no sugars put on them. Made those guys, purified them, injected them into The way this vaccine works, it's called the transmission blocking vaccine. So what you do is you inject the protein into a person, he makes the antibodies, then when the mosquito feeds on your blood, it takes up the blood and the parasite, and inside the gut, you duplicate copies of the parasite. Of the oocytes inside the individual. So here's untreated, you get lots of oocytes. These guys are quite infective. Here's the mouse injected with PFS-25. He makes a few oocytes, but not enough to infect the next person they bite. So that's why it's called the transmission blocking antigen or vaccine. And so that worked wonderfully, right? Okay, I'm just going to burn through two last things really quick, right? One is, so there are a number of things we can do inside the chloroplast that I think are unique, whether that's the malaria vaccines or the toxin or even feeding of the MAA. But ultimately, if you really want an industrial organism, you'd love to have proteins that can be secreted from the cells, and there's lots of stuff you'd like to do in the nuclear genome. Gene expression for a long time, because although we can get nuclear gene expression, very rapidly you get silencing inside of Chlamydomonas. And we don't know if this is going to hold up for other species of algae, but so far it looks like it is, okay? You get an advantage that you get bigger promoters, you can do fancier stuff, you can regulate them better, and as I said, you can target proteins, so you can either secrete them or target them to organelle, but you always get silencing. So one of the ways around this that other people have shown and we thought might work inside of algae is that what you do is you link that protein to the selectable marker. So in this case, we took an industrial enzyme called xylanase, and we link that to the bleomycin resistance. And bleomycin is a unique antibiotic because the way it works is by binding and sequestering. It's not an enzyme. So it doesn't bind the antibiotic and degrade it. It binds it and sequesters it. So the only way you get high levels of antibiotic resistance is if you have high levels of BLE production. So what we thought is, well, if we fuse the xylanase to BLE, do the transformation, select for high levels of bleomycin, we'll force BLE to be expressed at high levels. Those are the only transformants that will survive, and then we should get high xylanase expression. And sure enough, that's exactly what we get. So here's transformants where we have unselected. We put hygromycin in an unselected gene. We put in xylanase. Picked hundreds of these guys. These are just 6 of them right here. You get expression of those, but it's very low. The guys where we've linked the 2 of them together, you get about a 100-fold increase of the xylanase expression. And in fact, now we're up to a couple % of soluble protein. That's what's called industrial strength. That means it's commercially viable. The problem with that is that you now have your industrial enzyme or your protein, whatever it might be, a therapeutic protein, linked to a bleomycin protein. It's a fusion protein, and that doesn't help you very much because you're never going to inject a fusion protein into somebody, and if you're going to sell it as an industrial enzyme, people don't want the bleomycin on it. So there's a little peptide which comes from foot and mouth disease called 2A. It's 19 amino acids. And the theory behind the way this thing works is that you have a single gene, so you have a single coding sequence, and as the ribosome reads along, it gets to this sequence, and this sequence folds into such a way that it doesn't form the peptide bond between those last 2 amino acids, but it continues to translate. So you get one protein made, and then it skips, and then you get the other protein made. So you still have it linked genetically, so you still get the selection here, but now you have 2 individual proteins made. And people have shown in many eukaryotic system this works. We tried it in algae and it works great. Okay? If we have the unlinked guys, if we have the linked guy with no 2A sequence, bleomycin gets nuclear localized. So here's BLE-GFP. The 2 proteins are fused together. Bleomycin targets to the nucleus. We get a nice green nucleus. We put the 2A peptide between them so it cleaves. Bleomycin still goes to the nucleus, but now the GFP is free and it floats in the cytoplasm. So there it is, shows up right there. All right? So that looks like it works pretty well. And we said, okay, if that works, how many other fluorescent proteins can we make? All right? On our way to making a bunch of industrial enzymes, but how many others? So we just picked 6 of them. In fact, now we're up to 8 of them. And what this shows is we can make every single fluorescent protein imaginable. So we now have rainbow-colored algae. All right? So what is rainbow-colored algae worth? Right? Well, I don't know, except that I think if you can make rainbow-colored algae, you can make anything. Right? You know, and I'm sure these will be available in some store near you for, you know, Christmas or something. We'll get them to do something out of that. But the main thing is, it's just sort of an example that you can do anything. But more importantly, fluorescent proteins are a really powerful tool. right? And you can use them to do lots of things. So here we simply target them. So here's one of our fluorescent proteins. This happens to be called M-cherry. We put a nuclear localization signal on it. There it is in the nucleus. We put a mitochondrial sequence on it. There it is in the mitochondria. Here it is inside the chloroplast. Here it is in the endoplasmic reticulum. And then the real power of any photosynthetic organism, but certainly single cells, is can I do classical genetics with these things? So we took one fluorescent protein that we had labeled the ER and another strain where we had labeled the nucleus, and then we crossed the two of them together. And now here we have guys that have a blue nucleus and a red endoplasmic reticulum. And we have guys on the way that have yellow mitochondria and green flagella. So pretty soon we will have 4 or 5 different colors. But the power of this is what it shows you is that we can use both molecular genetics and traditional breeding to make industrial strains. All right? And in fact, we've now made one that secretes. So we took that same sequence. So here's the BLE, here's the 2A. And then after the 2A, we put a secretion signal. All right? So the ribosome comes along, skips right here. Now we've got a signal peptide that targets it to the endoplasmic reticulum. And now we have secreted xylanase outside the cell at industrial strength. Right? And we've got going everything you could imagine. Antibodies, erythropoietin, you name it. Okay? So I want to end in just the one last minute, right? Just on kind of forward thinking, what can we do next? So one of the things that we'd really like to do, we can make all of those industrial compounds, and I think they will have an enormous opportunity, as I said, because of efficiencies, because of photosynthesis. But what would really be nice is can we actually change photosynthesis, all right? That would be the real goal. Could we actually make it more efficient so we can make not only algae more efficient, but terrestrial crops, corn, soybean, you name it, all right? And I'm going to go through this quick just to show that we started with the reaction center proteins of photosystem 2. So we made a knockout strain in which we've deleted the photosystem 1 gene called PSVA, and then we put it back. This is from a higher plant, Arabidopsis. Here's a cyanobacteria. Here's a diatom. All 3 of those reaction center proteins express. So we could complement a knockout strain by bringing in genes from other organisms. I don't think too much of a surprise there. The real question is, could we go to organisms that have a different phenotype, meaning they do something different in photosynthesis? And when we introduce those new genes, can we change the phenotype of that organism? So cyanobacteria do something really well that green algae don't do, and that is that they respond to low light or high light growth conditions. All right? And in cyanobacteria, it's known, suspected why, because they have 2 different D1s. So one is called D1.1, and that's the low light form. So that protein, this isoform of the protein is expressed at high levels in low light. There's another one called D1.2, and that's expressed in high light. And people knew that if I grew cyanos in low light, I get more of this guy. And if I get more of this guy, it tends to transfer oxygen. As if we put the low light form into algae or the high light form into algae, it now acts exactly like the cyanobacteria, low light or high light grown form. But the more important one of that is what happens when we actually grow these guys under those conditions. So here is the green algae with a cyanobacterial gene put in it, which grown in low light between the high light form and the low light form, and the low light form outgrows the high light form in low light wonderfully. So we've imparted a new trait into this specifically from that gene. And then here's the high light form, and in fact it does even better because the low light form dies in bright light. So we can take a single gene, a single trait, and we can impart new function into this algae. Okay? Now the question is, can we do this with multi-gene traits? Right? Because many of you will know photosystem 2 reaction center is a lot of different proteins. Right? So this is the last slide which I'll show you, which is simply we went back and knocked out all 6 of the reaction center proteins from Chlamydomonas. We put those back from Arabidopsis, from Volvox, which is a closely related green algae, and from Chlammy, and all of those guys at least grow. We don't know yet, do they grow better, do they grow worse, but what we know is they're all photosynthetic, which means we can take an entire synthetic construct, all 6 genes, from one organism and move it into a different one. So true synthetic biology, right? Not designer organisms yet, but I think on the way, if we can figure out exactly which one of those proteins is conferring the trait. So what's the final frontier? You know, where do we have to go to next? I think a couple things. One is we have to get to industrial scale. This is a picture of the Sapphire facility. Down in Columbus, New Mexico, these ponds are 1 acre each. These ponds are 2 acres each. They have 100 acres growing now. They're doing this for biofuel production. They produced about 120 metric tons of algae this summer. They'll produce about twice that next summer. But the most important thing is what they're telling you is this is getting to agricultural scale. This is driving economies of scale now that we can start to think about producing industrial chemicals in this now and producing them on a scale that gives us agricultural costs. So many of you will know that nothing is cheaper than American agriculture. Nothing is, corn's getting more expensive, but it's still the cheapest commodity on the planet. And if we can do the same thing with algae and we can train it to make all these things, then I think we have enormous opportunity in the future. All right? So what are the challenges that we made? We have to continue to improve efficiencies. This is actually not so much of a biological problem anymore as it is a systems problem. So we actually have to get the engineers to come and start to help us think about these things. Because putting in the genes gives you something, but it's the entire system that gives you a product, right? Then obviously we have to reach world scale because only by reaching world-class scale do you really drive the efficiencies, right? And this has to be new production, right? It doesn't do us any good to go do this to corn. Algae gives us as an opportunity to do this in places where we didn't have agriculture and we didn't have productivity before. All right? We need to reach economic viability with a product. In other words, we have to sell stuff so we can stay alive. People always ask me, well, this all sounds good, but if it's good, why aren't there big companies doing this? I think the biggest impediment to that is because we simply haven't done it before. Part of that is because we didn't have to worry about it before because petroleum was so cheap, we could just make whatever we wanted chemically. You know, take that, convert it into sugars, and then ferment those sugars in yeast or something. But now we have to think about different ways to do it. And then finally, what I hope is that at some point we can start to improve terrestrial agriculture. Right? There's 7 billion people on the planet. We're not going to make it with the resources we have now. But if we can start to think outside the box a little bit and start to think about how we can improve, given this enormous genetic resource we have available, unlimited data, and now I think unlimited computation power, as I said, what it's going to be limited by is our imagination and how fast can we make these systems that actually produce something. All right? And I'll end it there, and thank you all for your attention. I'm sure there are a lot of questions. We've got microphones. So if you want to ask a question, please raise your hand. And James. Yeah, shout it out. Because we have remote people, and we need them. How easy is it on? How easy is it to domesticate, transform algae other than a standard? Yeah, so that is completely strain dependent. So on some of the industrial organisms that we're going to, which right now seem to be, they're going to be Nanochloropsis, Senodesmus, and a few others like that. Those have worked really well. Senodesmus, no problem at all, almost all of the technologies. Denaliella tertialecta. So every one of those, the strategies that we've used in Chlamydomonas work, but the DNA doesn't. So I cannot take the constructs that I made in Chlamydomonas and use those in Senodesmus or Denaliella. Those species are too different. But if I go and simply design the exact same thing using Senodesmus DNA or Denaliella DNA, so go cut out the promoters, cut out the UTRs, then they seem to work just fine. So I don't think that's going to be a big issue. It's going to be time, but that's it.
Hi, thanks. I really enjoyed the seminar. So in the couple of examples about protein production that you started with, the attraction to using algae was that it had a unique combination of ability to fold proteins using carbon chains isn't that a really different goal? And couldn't we just find a yeast that had this particular combination of organelle organization and glycosylation versus not?